Welcome to video 2.2. In this video, I'll be covering the structure of nucleic acids and discussing their hydrogen bonding. So introducing the nucleic acid, right, this looks something like this. So there's a few different parts, and to really, understand the building block, which is the nucleotide, I want to first look here, and then we'll discuss what the polymer looks like on the left side. A polymer is a string of multiple subunits, and what you're going to see, a big trend in biochemistry, is that we have some sort of building block, and then we make strings of that building block to make structures. This is true for DNA, and it's true for protein. The building block of DNA is the nucleic acid here, the nucleotide. So there are three key parts to this structure. The first is, we'll call it the sugar, right? This is the ribose sugar in the case of DNA or RNA, or the deoxyribose sugar in the case of DNA, right? DNA stands for deoxy, right? That's the D in DNA. We'll come back to where that, what that is in a little bit. But really, I want you to focus that this here, this five-membered sugar, this five-membered ring, that is the sugar part, the ribose sugar of the nucleotide. The second part is the base, right? If we can call it the base or the nucleic base. Right here, we see it connected to the sugar, right? And this is going to be a very important piece of information. You'll notice that they always tend to be nitrogen rings, and it is absolutely going to be important to memorize their structures. We'll talk about easier ways to make that less pure memorization and more patterns in a bit. And last but not least, the third part of the nucleotide is the phosphate. Right? As from the last video, we know that one of the unique things about DNA is that it has that phosphorus in it. A phosphate is when that phosphorus is connected to a bunch of oxygens. The phosphate is another important part of the DNA building block, and all three of these together make the nucleotide. We'll cover the naming in a little bit more detail, but if you remove the phosphorus, this turns into a nucleoside. This will be covered in a later video. So here we can see this nucleotide built here. And the way this works is that the oxygen down here, right, it can actually be connected to another phosphorus. If we do this, we can then connect it to another sugar, which is connected then to another, base. So I'll just copy the same base here. Right, so there's our DNA base, our nucleic acid base, and another OH. And so here what we've done is we've just put another one of these connected to the bottom, right? So that oxygen that was present in the original sugar, it can be used as a branching off point to connect another building block. It's like, clicking two Legos together. The same can be done up here, right, where the phosphorus is now, instead of being connected just to nothing, it's connected to the oxygen of another sugar. So we once again have another sugar. It's connected to its nucleic base that I'm too lazy to draw, and another phosphorus that I'm too lazy to draw. But you get the idea, right? We can make long chains polymers of these nucleic acids, branching off at this point and at this point. So with that in mind, let's go a little bit more into the structure of these, specifically the bases, I think, are the more important parts. It's important to note that the DNA sugar and the RNA sugar and the phosphate aren't really going to change much, but it's this DNA base that's going to have a lot of impact in the information carried by this material. So DNA versus RNA. DNA stands for deoxyribonucleic acid, so nucleic acid, and RNA is just ribonucleic acid. So deoxyribo and ribo. And that really gives you the first difference between the two. And an important, very crucial part of information is, here we have DNA and RNA. It's this OH here. So we actually number this sugar. Where it's connected to the base, we call it 1. This is 2, 3, 4, and 5. So this is the numbering of that core sugar. And you'll notice that that 2 position is really where the difference is. RNA has a 2' OH. 2' means that it's on the second carbon, carbon number 2. Where DNA, right, deoxy means remove oxygen. We remove the 2' OH. So we'll talk about the stability between these two molecules later, right? I think that's actually going to be in video 2.5. In video 2.5, you can jump there if you'd like. We'll talk about why this OH is so important for the stability difference of these molecules. For now, what I'll say is that DNA is very stable, and we need it to be stable, because what's its job, right? Back to the jobs of these chemicals. DNA's job is to ensure that we store information for long term, right? If we mutate the DNA, there's no other way to fix that, right? We need to store information for long terms. We don't want any mutations. We don't want any changes to this chemical. But for RNA, we actually want it. It's desired to be unstable, right? It's meant to carry a message, to print a protein, but we don't want that print a protein forever. We want that RNA to eventually degrade, and this OH is critical in that. So either way, that's the first difference that's really important to notice. If you're asked, is this chemical DNA or RNA, find that 2' position connected beside the nucleic base. Is there an oxygen there? If there isn't, it's DNA. If there is, it's RNA. Next is the bases used. So without drawing their full structure, I'll do that below. DNA uses 4 main bases, G, C, A, and T, for guanine, cytosine, adenine, and thymine. RNA uses the same base, right? Guanine, cytosine, and adenine, but there's a big difference with thymine and uracil. So the important idea is that RNA is never going to have a T in it. You're never going to see a T in the DNA sequence, or the RNA sequence, rather. And for DNA, you're never going to see a U. So if you're looking at the sequence, and maybe you're not given the actual structure, you can't see the OH. But if you're given the sequence and you see a U in it, you know that it must be RNA. And if you see a T in it, that it must be DNA. With that in mind, these are the 5 DNA bases that encode every piece of information, which is pretty crazy. So how does that structurally look? We're going to break down the bases here and talk about 2 classes. So the first class is the class of purines, and the second is that of pyrimidines. Purines are these based on their naming convention is from the actual ring they're from. This is called a purine ring. And it's got a lot of nitrogens. You can see there's, nitrogens, a couple rings put together, where pyrimidines are just a single nitrogen-based ring. And the DNA bases are synthesized from these starting materials. With guanine, this nitrogen on the top left here is going to be number 1 for the numbering. And you'll see here that with guanine, so that's the first one we're going to cover, you can see that base structure, although there are some differences, right? We see that 6-membered ring beside a 5-membered ring, as we did here. But for example, this nitrogen here, right, it's not double bonded anymore. It's single bonded and connected to the amide, right, by this. So there's an amide here. Right, so there are some differences, but for the most part, they look quite similar. So first, recognize that if there's 2 rings put together in the base, it's a purine, and your purines are either guanine or adenine. So 2 rings is your sign that it's a purine, and a big sign here is that guanine, right, has this carbonyl, adenine just has this nitrogen. So in terms of numbering, this can be quite odd, and I'm going to go through numbering these together. You'll note that this red hydrogen, this is actually where the DNA base will be connected to the sugar backbone, right? That ribose sugar backbone. So that's actually going to be nitrogen 9. And what we're going to do is almost draw an S shape over the molecule like this. So I like to imagine an S shape that ends up on that 9. So we have 1, 2, 3, 4, 5, 6. So we kind of go in a full circle around here, but then we swing back around. 7, 8, 9. That's how you can number those different positions. In molecules, right, find that where that, that nitrogen is linked to the sugar, the opposite corner is 1, right? That's a good way to handle the purines. Same thing for adenine. 1, 2, 3, 4, 5, 6, 7, 8, 9. Cool. With pyrimidines, right? We're going to see, again, small differences, but there's the big thing to, notice the difference between these two is that there's just one ring. If we look at cytosine, right, thymine and uracil now. So let's look at all three of these put together. They all have that same ring, right? The cytosine has a double bond and a nitrogen. Thymine has two double bonds, and so does uracil. Remember that thymine and uracil, these are the two to keep in mind. They're the ones that tell you if you're looking at DNA or RNA. They're very similar, but you'll notice that thymine, the only difference is it has an extra methyl group, right? In terms of numbering pyrimidines, it's more straightforward. They're going to connect on that bottom nitrogen, and where they connect is nitrogen 1. Then you just go around, right? Now you go around to the other nitrogen. So 1, 2, 3, 4, 5, 6. Again, you start at one nitrogen and you go to the other nitrogen, like so. So the nitrogens are always 1 and 3. With that in mind, let's talk about some tips for naming these, because this can be quite challenging. Obviously, these chemicals look very unique. But first of all, right, the number of rings is super helpful to determine them. So guanine and adenine are your only options if there's 2 rings. And then keep in mind that adenine is the one that doesn't have a double bond. There's no carbon-oxygen double bond, right? If you can keep in mind the whole 2 rings idea, and then remember that adenine is the one that has no carbonyl, you should be able to recognize and name these chemicals. For the pyrimidines, right, they all have 1 ring. If you have a methyl group, right, because that's the only methyl group in any of these structures, right? Again, a methyl group is just that one carbon kind of sticking out in nowhere. That means that you have to have thymine. Methyl equals thymine. Between cytosine and uracil, right, the big difference is, and you'll notice the only one here that actually has an NH2, right? So guanine and adenine both have NH2s, to be clear, but they have 2 rings. When you're looking at the 1 ring molecules, if you have an NH2, you're cytosine. And that means that if you don't have a methyl group and you don't have an NH2, that means you're uracil. So instead of trying to fully, and if you want to memorize the structures entirely with their numbering, you can do that. It's recommended. It'll take more time. But if your job is just to recognize them when drawn, keep in mind that you can just look for these landmarks, right? The number of rings and the presence of specific groups. All of these bases are unique in some way. With that in mind, let's move on to the concept of base pairing. So DNA is double-stranded, and bases can pair with each other, right? The idea here is that if I have one strand here with a G, and I have another strand with a G, these things are not going to want to link, right? There's this base complementarity where G's only pair with C's. So going back up here again, or I can just leave it here, this is guanine. I can tell it's guanine because guanine is a purine, and there are 2 rings here. This is cytosine. It's a pyrimidine. Now, guanine and cytosine have 3 hydrogen bonds. And now that we've covered hydrogen bonds in section 1, we can look at where these are. The first is one of these hydrogens interacting with this carbonyl. The second is the lone pair of this nitrogen interacting with this hydrogen. And the third is this hydrogen interacting with this oxygen. Right, so this nice complementarity in the structures, right, where again we have a donor here, and then, an acceptor here and a donor here and an acceptor, and a donor here and an acceptor, right? They're complementary, right? It's not acceptor, donor, donor, because if they were, donors wouldn't want to donate to each other and acceptors wouldn't want to accept from each other, right? G's and C's have this nice complementarity, this nice intermolecular interaction that keeps the strands together. And it's worth noting that because there's 3 bonds, they tend to be stronger, right? I didn't draw the other complementary things. It's not important. I don't think you need to be able to do this, by the way, but it's just nice to show, right? The other one to consider is the bonds in DNA between adenine and thymine, and in RNA between adenine and uracil, right? These both have 2 hydrogen bonds. So they're still connected, they're still complementary, but they are weaker, right? Complementary sequences is an idea or a concept where by looking at one strand and just imagining a second strand, we can think about what that sequence would need to be to totally match and to be complementary and to bind to the other piece of DNA. Right? If you want this strand of DNA to bind to this strand of DNA, it needs to be complementary. Since G's pair with C's, I'll write this down below here, G's pair with C's and A's pair with T's, or A's pair with U's, we can take a look at this. The first question is, this sequence we've drawn, is it DNA or RNA? The answer is that it's DNA, and we know that because we see T's, right? U is what we have in RNA, and T is what we have in DNA, so we know it's DNA. All we have to do is say, well, for G, this side would have to have a C, and then they would have 3 hydrogen bonds. C, this side would have to have a G, 3 hydrogen bonds, C, and we can just kind of go complementary the whole way. So there you go. This would be the complementary sequence that would bind this sequence, and we'll talk more about that in further videos. But this is how the structure of nucleic acids, the hydrogen bonding of those nucleic bases of both purines and pyrimidines, helps us understand the structure of these chemicals. Now, don't worry if you're a little confused, right? We're going to go back over in the next video and talk more about especially the structure of these nucleotides, breaking down their naming, understanding the different types of them and the structures they make, like the polymers, DNA, and RNA. I'll see you in the next video to discuss that topic.